In the previous lecture, we studied introduction of cell, its discovery, unicellular and multicellular organisms. Today, I shall be dealing with the structure of cell. As you can see, here is a structure of animal cell in which nucleus is there in the center. At the center, there is nucleus. Just outside the nucleus is endoplasmic reticulum. This structure is known as endoplasmic reticulum. Then, beside it, there is Golgi body, mitochondria, lysosome, vacuole. Then plasma membrane. We have plasma membrane, ribosomes, centrioles, etc. Now we will study about plasma membrane. This model is known as fluid mosaic model. It is known as fluid mosaic model. It is known as fluid mosaic model. Okay, and it was given by two scientists named Singer and Nicholson. Their name was Singer and Nicholson, Nichol. So it was given by Singer and Nicholson. It is a bilipid layer. As you can see on your screen, it is a bilipid layer. There are two layers in it. The first layer is this one and second layer is this one. Suppose it is the outer layer of the cell or outer part of the cell, and this is the internal part of the cell. Okay, then there are two layers in it. First one is the outer one, it is inner one. So it is known as bilipid layer. After this, it is made up of phospholipid. You can see here, it is made up of phospholipid. So what is phospholipid? It is a structure of this kind, this one. This structure, the upper one, it is known as it is called hydrophilic. The term hydrophilic means water loving. This side and this side there is water. Okay, the membrane which is in contact of water or membrane or plasma, it is known as hydrophilic end, and the membrane which is in contact with which is not in contact with water, this one, it is known as hydrophobic. It is known as hydro wait it is known as hydrophobic, which means fear from water. This is known as hydrophobic end. Apart from phospholipid, it has carrier proteins or proteins. There are two types of proteins in it. The first one is integral or internal protein, which is inside plasma membrane. It is called internal or integral protein. The protein which is to its external part, such as this one, it is known as peripheral protein or extrinsic protein. Okay, the function of protein, both the proteins, extrinsic as well as intrinsic, is to help in transport of ions and molecules. Is that clear? Okay, now we will discuss a most important phenomena, that is diffusion and osmosis. Plasma membrane is also called selective permeable because it gives permission to some of the selected ions or molecules. It does not give permission to every molecules. Okay, now we will discuss about diffusion and osmosis. So diffusion is basically the movement of molecules from higher to lower concentration. Suppose that here is a DO, okay, and here is its higher concentration, here is its lower concentration. Suppose a boy has applied DO on his clothes, then Here is higher concentration on his clothes and here is lower concentration in the surrounding area. So the molecules of the DO will move from higher to lower concentration. This is known as diffusion. So what is diffusion? The movement of molecules from a region of higher concentration to lower concentration is known as diffusion. Is that clear? You can see in the diagram that molecules are moving from higher to lower concentration. Clear? And if there is a membrane between higher and lower concentration like this, which is just like plasma membrane, which is selectively permeable, membrane is there and it is selective permeable. It is selective permeable. Then it is known as osmosis. What is osmosis? It is a process in which molecules move from higher concentration. They are moving from higher to lower. This is higher concentration, this is lower concentration. They are moving from higher to lower but across a semi-permeable membrane. It will not allow everything to pass over it. Is that clear? Okay. Now, there is one more topic in which hypertonic and isotonic solution is given. So I will describe you in detail about this, such as we are taking, suppose that it is a beaker, okay, and we have taken sugar solution in it. We have taken sugar solution in it, and after that we we have kept grapes in it. So what will happen here? Inside the grapes, water is high as compared to outer environment. Water is high, and in the sugar solution water is low, or the concentration of water is less. So in this case the molecules will move from a region of higher concentration to lower concentration. They will move from grapes into the liquid solution or sugar solution. Clear? This is known as hypertonic solution, in which the in which water moves from inside to outside. Basically you have to remember this example of sugar solution and grapes molecule. Okay. Now we will discuss another example, and suppose that we have taken there is a beaker. There is a beaker. It is filled with water. Okay, and we have taken gram seeds. Beaker, and we have taken gram seeds. So what will happen? Here is water. So in this case what will happen? The water molecules will move from the water is more in the beaker and it is less in the gram. So water will move from higher to lower concentration, from outer to inner surface. The water will move from beaker inside the gram. So this is known as hypotonic solution. Hypotonic. It is known as hypotonic solution. In this, water is also moving from higher to lower concentration, but it is moving inside the cell. Is that clear? And then third one is isotonic solution. For example, if we have a beaker, it is filled with water, and we are keeping a grapes molecule, or we are keeping grapes in it. Then what will happen? Here also we have sufficient water. Here also we have sufficient water. In both the cases, the concentration of water is high. In this case, neither water will go inside, or neither nor water will comes outside to it. So a type of solution in which water does not move neither inside neither nor outside. So it is known as isotonic solution. Is that clear? Now there is one more topic. We will start with the there are two terms, that is exoosmosis and endoosmosis. If a cell loses its water, if a cell loses its water, it is known as, here see they have written here, it is hypertonic solution. If a cell, if anything is moving, wait, I will tell you with the help of diagram. See here, exo and endocytosis. What is happening here? If when the molecules are moving outside the cell, it is known as exocytosis. See, in this case, water is moving, or any molecule is moving outside the cell. Okay, this is known as exocytosis. This one. And if water is moving inside the cell, see, it is moving inside the cell, it is known as endocytosis. In the same way, exoosmosis and endoosmosis can be explained. It is a process by which various organisms such as amoeba, paramecium, mostly all the single celled organism, they used to exchange their materials. Is that clear?